This is Kayleanna in the Fields Laboratory. What are you doing today, Kayleanna?
I am isolating RNA from MDMs, macrophages. And so first I am going to remove the top and remove all of the media just by pulling it up and putting it into a solution or a bucket that has a little bit of bleach in it.
And when you remove the media, you be careful. She's being very careful to not disrupt the cells, the monolayer that's on the bottom of the plate. So she's just putting the tip in the bottom of the well, pulling straight out and going straight into the bucket.
Yeah. And just doing one plate at a time, even though this experiment has 3 plates, because it's better to just isolate as much as you can handle.
Correct.
Which is about 12.
Yes. If you go over 12 or certainly over 18, you might end up leaving one sample longer than should be left. Okay, so now she's in the process of washing the PBS, washing the cells with PBS to remove any residual media that's remaining after she removed the media from the cells. And so she trips it down the side of the plate as the plate is angled. That way she's not disrupting the monolayer. And then once she's done, she will level the plate out to wash the cells with the PBS before removing it.
Like this.
Exactly. Now she's removing the PBS. She tilts the plate to pull up all the PBS. And this is the final stage before adding the RLT lysis buffer. At this point, after the cells are washed with PBS, she's going to add RLT lysis buffer that has been supplemented with beta-mercaptoethanol at a concentration of 10 microliters per milliliter. She'll add 350 microliters per well. And to ensure she gets all the cells, she disperses it from the top of the plate so it'll run down to the bottom of the wells. Being sure not to touch the bottom of the plate so that there's no contamination between wells. The cells are lysed as soon as the RLT touches the cells. And if you think you touch the cells and could have any contamination, you must change tips. In fact, it may be a good idea to change tips between each well. So the lysis buffer has been added to each well, and now it needs to be removed with the filter tip, changing a tip after each well, and placed into the gDNA eliminator spin column. Now that the lysis buffer has been added to the DNA eliminator spin column, you add it to the centrifuge, and you centrifuge at above 8,000 RPM for
30 seconds.
30 seconds. There it goes. So now the initial spin to get rid of the DNA has been completed. We will remove the insert that has the purple ring there, and to the solution that went through the gDNA eliminator meshwork, we will add 70% ethanol solution and mix it by pipetting up and down before going into the RNA spin column. So now she's taking from an aliquoted 70% ethanol in water solution and adding 350 microliters each tube. Now that the ethanol solution is added to each tube, she mixes with the lysate by pipetting up and down 3 to 4 times. And then she adds it to the RNA spin column. She'll do that with the remaining samples and then moves into the centrifuge. Then we remove the flow through into the waste bucket and put the RNA spin column back into the centrifuge. Now that Kayleana has removed the wash through and placed the mRNA spin columns back in the centrifuge, opening the cap, she's adding 700 microliters of RW1 buffer to each tube before another 15-second spin at greater than 8,000 Gs.
Use the short button.
Oh, really?
I always click this.
Oh. And then you wait till this gets above 8,000, and then you wait 15 seconds. Basically, you can wait until about 25 seconds, and that'll be fine. And then you move on to the RPE buffer. Now all the spinning is completed, and we're to the last step where we add water to the membrane so that we can elute the RNA from the spin column. And it will be collected in a 1.5 milliliter collection tube with the last centrifugation. So Kayleana gets 40 microliters of water, and she carefully places the tip right above the membrane so that the water drops directly on the membrane. Careful to not disrupt the membrane. Be sure and change tips each time.